Okay. All right. So, without further ado, I just wanted to say a warm welcome to everyone. So, this is our Inspire Me session. So, like all Panoramics Visions Inspire Me session, this session is where we invite speakers to share with us their journeys, their motivations behind their growth, and to kind of share their vision and also some of maybe some sort of advice to kind of guide all scientists. So today is basically it's a celebration of women in spatial, and this event came about basically at a conference where I was sitting and having a conversation with Dr. Trang Wu. And she suggested that, you know what, you should do something that has got to do with celebrating women in spatial and single cell. So here we are, and I'm going to hand it over to Alyona because, to be very frank, she's the one who designed everything and she's the one who planned the speakers and came up with this incredible program, and also with Ashley Willis too, who actually contributed to the design elements as well. So I'm going to hand it off, and we are really, really excited to have everyone here today. Thank you.
Thank you for a great introduction. We would like to welcome Dr. John Stratton as our keynote speaker for today. Dr. John Stratton is a neuroimmunologist at McGill University's Montreal Neurological Institute Hospital, where she co-leads the Neuroimmunological Diseases and Glia Biology Research Group. Her research leverages single-cell and spatial transcriptomic technologies to chart the cellular and molecular diversity of glial and border-associated cell types in health and disease. By integrating high-resolution multi-omics with human tissue and modeling systems, her group has revealed novel functions for glia and CNS border cells in neuroinflammation, neurodegeneration, and rare neurogenetic disorders. So, warm welcome for Dr. Stratton.
Thank you. What a privilege to be here. It's such a great initiative. I'm glad, Shamini, you had that conversation to plan this. It's really great. And there are a lot of females leading spatial and single cell biology. Some of the panelists here today will be really excited to hear the thoughts of everyone on how to manage the complexity of running an academic lab and having a family and all the things that us females do. So without further ado, I'm going to just spend the next 20 minutes or so talking about my training experience, what motivated me to do what I'm doing, and then hopefully share a few words of wisdom, at least things that I think helped guide me to where I am today. Okay, so to get started, I actually had no idea what I wanted to do in high school. I hope you resonate with that. I liked sports. I liked helping others. Biology interested me. My high school biology teacher was great. But around that time, my dad, who had multiple sclerosis, was getting more and more progressive. So he ended up being in wheelchair. And so I was witnessing this devastating neurodegenerative disease as a child, kind of growing up. But I didn't think I could do much about it. I didn't have that confidence that little me, interested in biology and sports, could really make an impact. And so life went on. I did an undergraduate degree in physiology. I did an honors year in muscle physiology where I learned how to hold a pipette and do a little bit of research. This was actually on an exchange year in Australia. I should have mentioned, but my dad moved to Australia when I was young, so I had been back and forth kind of co-living in Vancouver, Canada, and in Melbourne, Australia. But I think at that moment, I started to gain a little confidence in the sense of like, maybe I can make an impact in MS research. Maybe I can make a difference. And I think by getting a little bit of wet lab experience during my honors year in a muscle physiology lab, that kind of helped. And the other thing that helped a lot is my boyfriend at the time, who's now my husband, and we have two children, was like the most supportive thing in the world. He came with me to the open days at University of Melbourne, where I ended up meeting the lab that I ended up joining as a PhD student, and that's kind of where it all started, in the sense that this lab was an MS research lab, and they realized that the bit of physiology background I had in the wet lab and my passion for MS research, that I might be a good PhD student. And through that experience, I had 5 years of doing what I was hoping to be mainly MS research. That was the goal. But as I'll explain to you a little bit later, I've had a lot of pivots in my career, and I really ended up in a field that was completely unexpected during my PhD that had nothing to do with MS research, lo and behold. And I'll talk to you about that in just a sec. But at the same time, I was starting to gain confidence beyond research. I was realizing I liked leadership positions. I liked making a difference in people's lives. So I founded a society called Students of Brain Research, SOBR. Thought that was a pretty cool name at the time when I came up with it, where we helped support any brain research student wanting to have professional development experiences, to realize their potential in leadership positions, and help them with symposia organizing, and help people, I guess, realize their full potential. And so I was doing research, now not in MS research, but I was doing a lot of extracurriculars that I was really passionate about. And that led me to a postdoc position now back in Canada at the Hotchkiss Brain Institute. Again, not in MS research, because my PhD didn't end up being MS research. And so my skill set ended up being in a completely different system, the nervous system of the periphery, not the central nervous system where MS affects. And this was the only position I could get because of my background. So the back of my mind, I want to do MS research, but I can't because I don't have the right technical skills for MS research. So I really, again, and I'll show you in a second, had to pivot at this point as well, which I'll talk about in the next slide. But again, at this moment in my career, I was still really engaged with extracurricular activities. I did a TEDx talk in Calgary. I was writing children's books. I was publishing a lot. In my postdoc, I realized the importance of publishing, publish or perish. I really gained a lot more confidence to really lead my own research, even if it wasn't MS research. And at the same time, we had a family. So my husband and I were married at this point, and we had our first child, little Sammy there. And during the first 6 weeks of her life, I flew over to Montreal, and I said to myself, I'm going to at least try to interview for independent academic positions, because at that point I had a pretty strong CV. And when I came over, actually, I met Karina Nagy for the first time. She was a partner in crime, she's on the panel as well, and she was one of the only other people doing single cell transcriptomics in the brain in Canada at that time. So we met. I ended up getting the job. I interviewed well. I guess they were impressed with a mother coming over with their baby literally in hand and doing breastfeeding sessions during the interview. Uh, no. So I got the position. I said, the philosophy at that moment was, I'm going to give it a shot. Um, and it was a position in multiple sclerosis research. So my entire training was not MS, and now finally I am founding my lab 10 years later in MS research. So never lose hope of your dreams would be my first point, to really emphasize. And I found a way to go back to my roots, and it doesn't mean my 10 year of training was not wasted. I learned a lot. I got into single cell technologies. I did some really important research in peripheral nerves. But I was still able to keep a fire lit at the back of my mind to get back to where I really wanted to be in the first place. And just a little bit about my lab. I think it's important as an independent faculty member, and I hope if you're interested in that, that's where you'll go one day, is to really focus on team building and funding opportunities. So we have a lot of great foundations that support us. The MS Canada. Here's a philanthropic donation from Le Grand Portage. And so I think a lot of what I do now, and it's been 6 years since I started my lab, is really cultivating that strong collaborative community within the research environment and sourcing funds, getting funding for the cool research that we're doing. And at the same time, this is during COVID. We had a COVID baby, 2021. We had our second child. And so this was a huge part of our lives. It still is. My daughter is now 7 and my son is 4. And so it's constant balancing act. My husband's a busy guy too. He's got a career in sustainability, and we're constantly managing family, work, life balance. And I'm sure we'll talk more about that after. So I wanted to talk to you about my challenges. So this idea of pivoting, this idea of me not doing MS research for 10 years. I'll tell you why. I started in this lab. It was a great MS research lab in Melbourne. Tobias Merson, my supervisor, was very supportive, and he had this great model. It's a transgenic system where you could specifically kill the cell types that are affected in MS. This is the oligodendrocyte. And so I supported a lot of research to characterize this model and learned a lot about this model. But what happened is when I started working on this one unique line that we thought had the best phenotype ever for studying MS research, I checked everywhere in the brain, and I'm like, there's nothing happening in the brain. What is going on with this model? It's not MS at all. And then finally, just by chance, I checked in the peripheral nervous system, and I could see the transgene that we were working with was actually very much expressed in the peripheral nervous system. And so I had this model. I was distraught that I could study peripheral nervous system dynamics. I could study demyelination and remyelination, all the things I wanted to do in the central nervous system. I could now do in the peripheral nervous system. So I had to re-gear. Within a few years, I was now the expert in my lab on peripheral nervous system biology. No one else knew a thing about it except for me. And that was a huge learning experience for me. And the idea of adapting to still have success in this new field was a big moment in my life, probably one of the biggest moments, with the challenges in my life. And I still look back now thinking, I felt like it was gonna, kill me, but now it's only made me stronger, and it actually was a really transformative learning experience for how I do research now. And so I mentioned already that, through this experience, I still was successful enough to get a postdoctoral research position back in Calgary on peripheral nerve. And so we looked at how macrophages, the immune system, regulates the myelinating cell of the periphery, Schwann cells, and published some nice and very important work in this field. But back to the idea of pivoting, at the same time, my supervisor, Jeff Bernaski, who was a biologist and regenerative medicine, not only in nerve regeneration, but also in skin regeneration, noticed something about a model he was working in. So this is a hair follicle. He works in hair and skin regeneration, and he noticed that the fluorescent signal that he was using to characterize this hair follicle regeneration was also expressed in the brain. And so he knew my history, that I wanted to do CNS research. I wanted to work on the brain, but I was currently working on the nerve. And he said, check out these mice, check out these brains, go with it, do what you want. I was very lucky to have a very open supervisor that allowed me to take things in any direction I wanted to as a postdoc. And so I got an undergrad student, Prajay Shah, to work with me on this side project. And the side project led us to basically publish in Cell. And so sometimes where you least expect success is where the success is. So the idea of adapting and pivoting to this peripheral project that in theory was just a random observation my supervisor made at the time, it can pay off. And it was right around the time when single cell transcriptomics was getting big. So I was able to process the cells that we were interested in, which are these green cells called ependymal cells, in order to get their genetic signature and characterize these cells and pretty much flip a dogma on its head for what these cells can and cannot do in the brain. And so this was helpful, but it didn't get me back to MS research. What are these ependymal cells really in the context of MS? My supervisor at that time didn't really care about MS and barely cared about ependymal cells, but had a cool model. And so what I had to do then is convince the people who hired me in an academic position, my own independent academic position, that these cells matter in MS. So I had to really merge fields, MS research with ependymal cell biology, and figure out a niche or an angle that was very unique. And of course, do this with some great techniques like single cell transcriptomics. And so my, I guess, conclusion when I reflect on my training experience is follow your nose, keep reading deeply because you'll make connections that maybe no one else has before, and be engaged and adaptable. And I think these are all things I did, and it kind of has led to me where I am today, and is definitely still shaping how I do research in my own lab. So I just want to talk a little bit about single cell in a little more detail because that's why we're all here. We all believe in single cell, I hope. So in the era of big data, things have shifted a lot. I was very lucky, and I do think for me, a lot of my success is being at the right place at the right time. And so in my PhD, this is right around when the big data era was beginning. The changes in how we store data, manage data, access data. We're in the digital age now. And then getting into my postdoc, that's right when omics was getting big. Before that, it was microarrays. And so I was aware of this change in the field, where we were and where we were going. Now, of course, with machine learning and AI, it's just getting bigger and bigger. So I was lucky to kind of have that awareness, I guess, during my training. And just to illustrate this in another way, the Human Genome Project took 13 years to achieve. The same sequencing now with the next generation sequencing approach takes the matter of a lunch hour. So it's completely changed how we do, or the speed and the cost at which how we do omics. And that's really allowed for single cell genomics to be such a big field right now. And it's only growing. So the fact that you're even on this call right now and you're interested in single cell, there's such a future for you in this space. And it's just a matter of figuring out what your question is and how you want to apply this really important technique. To illustrate this yet again, there's a reason why Nature Methods has had single cell transcriptomics, multiomics, spatial, all in the last, 5 years or since 2013 at least, this idea that it really is the future of how we can do biology and understand our complex organisms. So one thing that's shaped me, because my background is biology, neuroscience, muscle physiology. I've always constantly been asking myself, if we're going to use these techniques, which are so powerful, we want to use them in the right way. We want to use them to truly advance our understanding of a mechanism, perhaps treat a disease like multiple sclerosis. And I think what happens sometimes is we end up focusing on the fact that it's so exciting that it's a new technique, and let's just throw everything at it and create a bunch of atlases, which is a great resource for the field. But I am worried sometimes that my lab just gets stuck down that path. And so I'm always thinking carefully about how can I balance that kind of direction. I'm excited about the technology with staying true to my roots, which is how do I actually understand multiple sclerosis in this case in order to better treat this disease. And so, every time I have these moments of reflection, I'm thinking about the different approaches that we use in single cell. Now I'm looking at gene regulatory networks using the most advanced algorithms and attack multi-omic sequencing. We look at cell-cell interactions to understand how the immune cells in MS are interacting with those ependymal cells that I spoke about before. We look at the organization of how these ependymal cells are in close proximity to some of the damage we see in MS brains. So we're finding ways to really leverage this technology, and at the same time in the lab developing new approaches. So we're now focused more on single cell proteomics to try to get that method working, which is challenging because it's not as developed as some of these transcriptomics approaches. Um, and along the way, we're also collaborating a lot. So just to share a few successes over the last 6 years, we've been working with other PIs on their research questions. We've been focusing, of course, our own. I've now started doing Parkinson's research, which I wasn't expecting as a result of having access to these really advanced techniques. So I think anything you do with this technique, you're going to find success because there's so many people who need and want to use this technique. And so publishing in this space and getting a name for yourself in this space as a trainee right now, it's really going to help you long into the future. But one thing I've learned as a PI now is writing is so important. So the sooner you start to learn how to write, formulate your ideas on grants, writing papers, the better off you'll be as you become independent. Because to get grants, you really need to convince others through writing that you're the one to do the research that you're describing. And your peers will be reviewing it against others and judging you on that. So I can't emphasize enough, in addition to doing great single cell work, that you need to really focus on writing skills as you mature as a scientist. And part of writing, I think, that I would really encourage you to think about is the conceptualization, right? I mean, now you can ask ChatGPT, please fix my sentence structure. You know, that's fine. You can have someone else help you with your grammar and your sentence structure. But abstract thinking and conceptualization of complex ideas in our field with single cell as your solution, your tool, putting that all together as one, it requires a human. AI can't do that yet. So please, when you're thinking about um, how to put a concept together. Imagination is key. I think you really need to be able to conceptualize things and discuss things with your peers and think outside the box and be curious. Every observation you make, it might be the thing on the periphery that you weren't expecting. Don't ignore that. That could be something interesting, and constantly refer back to literature to try to make sense of it all, and don't be afraid to fail, because maybe 10 of your ideas are a bunch of crap, but that one, that's the change maker. That's the one that's going to really change your field. So it's okay to have difficulties and challenges, because that's, I think, where the most success or opportunity might lie. Okay, so I'm just going to finish on last few um, comments about, I guess, where we're going with single cell, in my opinion. So, the field right now is already at about $6 billion across the world. So single cell's big. And the logic is it will only ever grow. And the reason for that is people hopefully will be able to use it more effectively, and will be able to extend this beyond our world of research biologists, bioinformaticians, and into the clinical field, into industry. And, at some point, of course, this is all impacting the general public because we're treating disease, and we should have the general public also be able to engage with with this technology, because it has a lot of power, and we need to explain that power and keep funding the kind of work we do. And so, one thing that we've been focusing on that we think is going to help this transition from keeping this inner circle to groups like ours into the larger field is making things more accessible. And to do that, we've developed a website. So you can see some of the websites currently that exist where you can display single cell data, for example, CellX, Trailmaker, single cell portal. But we've recently developed something called SingloCell, which we hope, and please check it out and let me know if it's true, is just that little more user friendly, a little more open, a little more easy for a clinician or someone in a clinical trial to check whether the single cell that they were looking at is changing with the treatment they're using. And so I could just show you a few plots. The logic is that you can circle or grab any of the cells, the single cells that you may want to be assessing, compare that between disease and control, let's say. You can then check what's the genes that are changing. We have now applications to look at spatial data sets using the same single cell website. And finally, we're now getting others to upload their own data set. So if you have single cell data and you want it to be open to the world for anyone to be able to view and play with, you can actually upload it onto this website. So I wanted to mention that just as another tool for everyone on the call, if you're interested, because we really do want to get more people engaged with single cell. And the last thing I'll say is everything we're doing in the lab, including the single cell website, is really made possible because of the team. So, we have had a lot of different people in the lab. Some people, web developers, like the person who's making the single cell website, others on the wet lab, and some on the dry lab. We really need everyone. It's a really diverse team where everyone matters. Everyone has a role. And the challenge for me is to make sure we're all speaking the same language. So when there's a computational biologist trying to explain the nuances of their code, that a wet lab person can understand that and help problem solve together. So I really see my lab as a collective brain that we all need to feed into to really truly get the most out of big data, single cell, to answer our biological questions. And with that, I think there's one little thing I'll end with, and this is just a little quote that I love, and I don't know where to put it, but I think if you're ever stuck and you're not sure and you don't have a strong urge in any specific direction, what I would say focus on, maybe in your PhD, sometimes this comes up, just be passionate and dedicated to pursuit of short-term goals. Be micro ambitious, put your head down and work hard with whatever is in front of you at that moment. You may not figure out your exact purpose in life right away, but chances are, if you're working hard on something that's important at that moment, there's probably going to have success, and keep your eyes open because some of that success might be somewhere you were least expecting, which is definitely the case for me. So thank you all for listening, and happy to take questions.
Thank you so much for a wonderful, wonderful presentation, Dr. Stratton. Very, very inspirational journey. We'll now open the floor up to the audience for any questions. So you can either raise your hand and then unmute yourself, introduce yourself, and ask the question directly, or you can also type it in the chat. I think there is a question from Stephanie.
Hi. So thank you very much for this very inspiring talk. And I think I wanted to say the single cell, that sounds like a really great initiative, because spatial single cell biology is the future. And I think this is definitely a technology what also the broader public should really know about.
Mhm.
But my actual question was, how do you best learn to write? Because at university, there are often no courses, and I have the feeling just reading papers does not help. And
Yeah,
yeah.
that's a great question. So I definitely was not a strong writer. When I was in my PhD, I never was taught in undergrad how to critically think and conceptualize, I guess a logical argument. And I think that should be taught in undergrad nowadays. I think it's unfortunate that we don't have classes that are solely dedicated to that, because that's the perfect environment to start to learn. So I really learned it by having in my graduate studies, having really great supervisors. So every time I wrote something bad, which was most of the time, they corrected it and they kept track changes on. And I I have to say I do this for my students now, and I wonder if how many people accept all and just move on with the track changes versus those that actually look at each individual change. What I did is I looked at each individual change. So I truly went through each sentence and I was like, why did they say that? Why did they do that? I really reflected each time I was getting corrected. I have to think that helped because now as a postdoc, I was really able to write all my papers on my own. My my supervisor would just do a quick little brief over, a few little minor edits, and it was kind of done. So I think that's where I learned. And I know you said, oh, papers, read them, read them, read them. It's true. People say that. Just read papers, you'll learn. There is some element of the more reading you do, the more it kind of you don't. It's subconscious. It's kind of in the back of your mind. There's this certain formula on how to write papers that when you read more papers, you start to see that formula. And so I would keep reading and I would find somebody that is willing to really correct your work. And so find a good mentor, I'd say is probably the advice I'd give. Mm,
Thank you.
no. Oh, two questions. Uh, I don't know who was first. One of you guys. Was it Karina?
I actually, it's not a question. I just wanted to add to what Joe was saying because I think, she was sort of saying this, but I'm going to be explicit about it. I think that when you are writing, you need to be intentional with your words. So it means that every sentence, every word in the sentence has an intention. And that's something that, when you're more of a creative writer, maybe some of them are in there for whatever reason, but in scientific writing, you really want to make sure that every word has an intention and that you're using it correctly. And I think it takes time, but those are the types of things that I think Joe and I at this point are correcting in our students, and we're hoping that you're catching that correction because sometimes words are put in there and from our perspective, because of our experience, we're like, why? So maybe that's one thing to think about when you're writing. So
Yeah, it's great. Very true.
Hi, Dr. Jen. I want to take you back a little. So I'm just interested in the single saw.
Yes.
I wanted to ask, does it only analyze data or does it come with sample processing? Because I'm thinking if you are to use it to monitor treatment over time, does that mean you have to do sequencing over time? Like that
Yeah.
might be a bit ethic.
Yeah, that's a great question. So, um, the other website that I mentioned, one of them is called Trailmaker,
Okay.
and there's another one. What's it called? It'll come to me in a sec, which is dedicated to actually the early processing steps, which means right off of the sequencer, you got to do your alignment, you got to do some QC. Our website does not do that. It's really important. And there's a reason for that. It's that we want to make sure each person who's putting data on the website has a chance to really go through their data because the QC steps can be, complex. And we can't be sure exactly how to do it for each individual data set. So unfortunately, you still kind of do need a bioinformatician level person to kind of go through and filter it and make sure it is what you want it to be. So our goal is once you're at that moment, then of course you can make it public because you're confident in the data at that point. You know what you want, you're happy with the data, and that's kind of where we come in with the website. But to your point about the idea of monitoring disease progression, like longitudinal data sets, we can definitely put that on our website, but you'd have to do all the QC and alignments and stuff first, and then put it in as one object. But we need tools like the one you're saying. We need clinical trials to have, a very quick, and it sounds all QC'd the same, and the samples are done by the same lab, all the same methods. you should and you can, with some of these Trailmaker like websites, have a fairly quick turnaround for aligning. And I think 10X might even have one now. Is that right? I don't know if they I think they were planning on developing one. Adriana might know, to speed that step up, because it is a bottleneck. I think that step, because you need server space, right? These are massive data sets that you need,
Thank
Yeah.
you.
My pleasure.
Thank you so much. So we'll take one more question, and we can save the rest for our panel discussion. Thank you.
Eden? Yeah.
Hi, sorry for the background noise. Thank you for the great talks. Was such inspirational. Um, I have one question. I work in a Center of Advanced Single Cell Analysis at Kids Hospital, and then my question is, what's on your input, like how to keep up with the new technologies, like developing every time? And then, all those new technologies are not cheap too. They're expensive with the funding and everything. Like, what's your input in this matter? Thank you.
Yeah, that's a great question. And it kind of comes back to one of the points I was making, where I'm constantly reflecting on what is my lab? Is it a biology lab where I use stock standard single cell approaches that are proven to work and just kind of have my pipeline that works? And for certain projects, that's all I need. So it's fine. But then there's this longing that I want to be, involved with the best and the latest revolutions in the field, because they're going to keep coming. And so why stop myself? Why limit myself? So I don't know if I have the right answer. I mean, obviously, I'm up to date with all the preprints that come out. Like, you can set your um your Google alerts and stuff to make sure you're aware of papers that come out that are on preprint servers. You'll definitely at least be aware of the papers, which is good. But whether you choose to implement, you have to write a grant on it. You have to get the collaboration usually, because someone else maybe developed the method that you want to now apply. So there's a lot of steps to actually getting it to be one of your, pipelines within your lab that you'll take. And so the only one I've really done that with is single cell proteomics, because I'm just really passionate about it. And so I guess I've just taken one small slice of the pie in that sense, but I wouldn't really call myself a tech dev lab. Maybe Karina's lab's a little more in that space, but it's tough. If we had more of a methods development person on the call, I think they'd probably be able to comment better than me. Yeah.
Thank you so much. And thanks again, Dr. Stranden, for your talk. We will now proceed to our career journey talks. That will be followed by a panel discussion.
Thank you. Thanks, guys.
Thanks again, Joanne. That was really great. So yeah, I'm really excited, and it's an absolute pleasure to introduce someone that I know very well because we work in the same lab, and that's Dr. Shruti Purushothaman. So Shruti is a postdoctoral fellow here at UBC in Dr. Frida Miller's lab. She is a developmental and regenerative biologist, and she uses single cell and spatial transcriptomic methods to study the regenerative processes in the digit tip of the mice, so the very end of their little fingertips. So it's really cool work that she does. She's currently funded by Michael Smith Health Research BC. She's very passionate about mentorship, science communication, and outreach. And I wanted to say that beyond science, Shruti practices Taekwondo. She is an incredible dancer, and she has a very, very, very adorable son. So thank you very much, Shruti, for joining us.
I'm trying to share my screen. Okay, can everyone see this in presenter mode?
Yeah, we can see it.
Okay. All right. Thank you, Ashley, for that wonderful, wonderful introduction. And Thank you, Panoramics team, for having me share my journey on complex tissue regeneration. And I did not realize how long I have been working on complex tissue regeneration till I actually made this slide. It's been quite more than a decade now. So I come from India, a small state, a South Indian state called Kerala. And I'll start with my major research experience that I had. It was mostly in my master's from Kerala. And then I moved to another state up here, Telangana, where I had a summer training program in the Center for Cellular and Molecular Biology. I was assigned to this regeneration biology lab that used multiple species, vertebrate and invertebrate species, to understand appendage regeneration. So I was involved in multiple studies in this lab, but my main focus was on complex tissue regeneration. Some of the examples being this is Amphiura. It's an echinoderm species which can regenerate its arms, and then caudal fin regeneration in zebrafish. So all this was happening around in 2012, 2013 time when I was really active in the lab, and we were trying to analyze what genes and proteins are expressed during the different regenerating time points. And of course, single cell techniques were not available back then in India. So what we went on to do was the smoothie kind of approach, using bulk RNA transcriptomics, some label quantitative proteomics, and then finding gene regulation for certain targets that we found interesting in previous papers using chromatin immunoprecipitation. So while all this was happening, I started thinking about going for grad school. And one thing that I really, really knew that I did not want to compromise on the field of study. I wanted to do appendage regeneration and possibly a lab that uses multiple model organism to do that. So that was a challenge. But luckily, I found home at the Seifert Lab, which is at the University of Kentucky. So I flew all the way from India to America all alone in my mid-20s, which fortunately turned out to be really good for my career so far. So what attracted me to Ashley's lab was that he was using multiple species, like the African spiny mouse and axolotls to understand complex tissue regeneration. I was really fascinated by axolotl limb regeneration, so I chose that as my projects. Although my focus was on limb regeneration, I ended up doing a limb development thesis that can be found online here. So externally, both these processes look very similar, and at single cell level, people have found that some of the circuitries are also overlapping in both these. And there was also a scarcity in the knowledge that we had about axolotl limb development. So I chose to proceed with that project. So everything that we knew up until then about limb development was based on these other model species which did not regenerate their limb. So that became really interesting to me. How does a regenerating species like axolotl develop the structure in the first place? So my primary focus was on understanding FGF in single cell sonic hedgehog signaling in this particular model. So I ended up doing a lot of whole mount in situ hybridization throughout my PhD. Back in 2014, when I started, axolotl genome was not published. So it was a really big struggle to find out the sequences from the labs that were sequencing the axolotl genome and get those sequences, make the RNA probes from scratch. It would take a week or so to make one probe and do the classic in vitro reverse transcription and get the probe ready in the lab. So now when I'm looking at the Xenium panels, there are panels available for axolotls too, I guess. Visium panel at least is available. But again, that's how the technology has grown, and I'm happy for the students now. But everything, all those are very expensive. And for beginner labs, I think this is still a great way to make your probes in the lab if you have a certain number of genes that you're interested in. All right, so during my PhD, that's what I did. I focused on axolotls and understood that they'll develop their limbs a little differently compared to all the more advanced species that do not regenerate their limb. But my interest was to find out why us as humans or a mammalian model like mouse does not regenerate the entire limb. And around the same time, I had a tragic personal experience. My mom had undergone a limb amputation because of diabetic gangrene, and this made it really, really important for me to focus on this area of study. And I've put this flyer for my dissertation seminar. And if you look at the date, it's 2020 November, which was peak COVID. And at that time, I was not even sure if I could graduate and find a job. But things really worked out for me magically. I found a job, which I'll talk about next. But I really think that what helped me was what all the preparation that I had done before COVID and the mentoring I got from Dr. Ashley Seifert in terms of writing, in terms of presenting my data set, that really helped me during this tough time to find a job. So I moved from the US to Canada to join Dr. Frida Miller and David Kaplan's lab at Sick Kids. Frida has now moved to Michael Smith Laboratory at UBC, where we work now. So they almost had the same questions about how progenitors would behave during development and regeneration. And more than that, I was even happy to join Frida because I've never worked under a woman scientist before. And I really was looking forward to learn her experience because she's been really successful over the past years in the field. And it's been really amazing to have her as a mentor. So this is the model that we work on, the mouse digit tip. And depending on the injury where we make, you can study this model for both regeneration and fibrosis. So if you make an amputation here, sparing some of the nail-associated tissue, the structure regenerates in a couple of weeks. If you make another injury, which gets rid of all of the nail-associated tissue, it results in fibrosis. So this is a very cool illustration that I always show, especially to a public who is not really familiar with this field. So this was all this illustration and a bunch of videos were made in collaboration with the UBC Biomedical Visualization and Communications program in the first year of my postdoc, which was a really fun thing that I did before I started the real serious work. Okay, so when I went into Frida's lab, we had this enormous amount of single cell data sets, which was published in DevCell in 2020. And what we did later in the next 2 years was to pick out just the, specifically look at the mesenchymal cells that are within this regenerating region. And this is here a light sheet video that you can see marking these nail mesenchyme cells. And we found that they are really important for regeneration. So that got us the cell reports paper even before I finished my second year. So that was a lot of work. And then I took a break. I had my son in early 2023, and I went on a maternity leave for 6 months. So I'm a kind of person that I do not like to not work. I just like to be active all the time. So I was not missing out on anything because the Xenium technology was developing within the lab around that time. So the neuroscience people, including Ashley, they were setting up the Xenium protocols in the lab. And in the very recent times, we've had some beautiful papers come out on spatial transcriptomics using Xenium from Frida Miller's lab. So by the end of my maternity leave, all these protocols had been set. And here my husband, who is also my colleague, he was helping me with setting up the Xenium protocol for the digitives. So that's what we did next. We were really interested in knowing the spatial patterns of the different cell types and the ligands that were expressed during regeneration and fibrosis. So we established a xenium-based protocol for the digit tip regeneration model here, which is a paper that is in prep right now and hopefully will come out soon next year. We are also using xenium to identify the cell types that are different in regenerating and fibrosis conditions, which is also a part of this paper done in collaboration with Sandeep Saxena, who is a postdoc in the Miller Lab. All right, so to sum it up, I've started my journey in India and then moved to the US and then Canada within the last 10 years or so. There has been an increase in the complexity of the model organisms that I've been using, from invertebrates to fish, amphibians, and now mammalian model. There's also been a increase in complexity in the approaches that I take, from bulk transcriptomics and proteomics to spatial gene analysis of one gene at a time using in situs. And then here there was single cell transcriptomics, single cell spatial transcriptomics, and a brand new model, which is the mouse, and molecular transgenic, of course. So that's been this last leg has been a really steep learning curve. But as Ashley said, I keep dancing. Whenever challenges are thrown at me, or even just for fun. So that's very helpful. And finally, coming to the end, I'd like to acknowledge a bunch of people. Ashley Seifert, my PhD mentor, he took me in when I did not have any publications. He just read my master's thesis and gave me a chance and mentored me really well in terms of writing, in terms of presenting my data, and also he's a really good friend. Frida Miller and David Kaplan, they've been really patient because it was a steep learning curve for me. They've been so supportive throughout the journey of my postdoc. And of course, all Miller Lab members for their support and the friendly moments that we have. Finally, all the funding agencies who've supported my research through grad school and postdoc. Thank you, and I'd be happy to take questions.
Thank you so much for your talk. Very inspiring journey as well, and very unique.
Thank
Everything. I think everyone has their own path, and it's so interesting to hear about how you got to where you are now. I think we have a time for one question. If the audience would like to ask something, if not, we'll just move on to the next talk and we'll take more questions at the panel discussion. All right, I think we're good to go. Next up, we would like to introduce Dr. Nagy, who heads the lab for neurogenomic exploration for underlying sex differences, Nexus, and is an assistant professor of psychiatry at McGill. She's an early career researcher at the Douglas Research Center. She is also a principal investigator at Ludmer Center for Neuroinformatic and Mental Health and a co-leader of HBHL Initiative for Single-Cell Neurobiology. She has extensive expertise in technology integration and was an early adopter of single-cell technologies, developing protocols to isolate high-quality nuclei from post-mortem brain for single nucleus transcriptomics, resulting in the first and largest single-cell dataset for MDD.
Okay.
Thank you.
Oh, thank you very much for the introduction. Is the correct slide showing? You're seeing the presentation and not the...
Yeah.
Thank you. So thank you so much for this invitation, and thank you very much for that introduction. So I'm here to tell you a story about my journey, and it involves sports, neuroscience, psychology, molecular biology, genomics, and being a woman. But first, I'd like to show you some stats. So back in the 1970s, there were not so many females with academic positions. And we've made great leaps and bounds in this category. In the early 2000s, we certainly have occupied more of these positions, but as you can see from the red, we're not really occupying the leadership roles. And still, this is from 2022, and this is from Canada, I have to say. So this is not necessarily the same around all countries, but we are reaching numbers, but we're still not reaching the leadership roles. So I think it's important to put the context to the journeys of women in science in general. But my story is just one of 2.6 million women in STEM worldwide, and I think that it might represent some common threads that a lot of us have to deal with, and they have two key ingredients. And those ingredients are resilience with a pinch of luck. So it started well back in my undergrad, and I was basically trying to figure out where I wanted to go. And kind of like Joe, I was very much into sports, and I really wanted to make sure that I could continue my athletic career. So it was important to me to find a place that could support that. And McGill not only had a long history in neuroscience, but it also had a strong program for the women's soccer team. So I found myself choosing McGill for that reason. I didn't exactly know what I wanted to be either. I could be an oncologist. My father had cancer, and I thought maybe that's the direction I wanted to go in, or maybe I wanted to go into psychiatry. Who knew? But when I came and I tried out for the varsity soccer team, I got lucky. I was able to make that team. I had a full course load. I actually had to work because I wasn't in a position where anybody could pay for me, so I needed to pay for my living costs and my tuition. And all was well until I broke my ankle. So the dream that I had sort of set out to do, the plans that I had were kind of kiboshed by this whole incident. And when this happened, I had a physiotherapist who told me, you know what, you need to stop doing high impact sports. And I didn't really understand what that meant, but they also said, why don't you try rowing? And I thought, okay, why not? This should be easy. I've done a lot of sports. I can do rowing. And it was by far the hardest sport I ever had to do. And it was another community, and it was an interesting one, and I was very grateful to be part of it. But I do recall being told at one day after a race that even though it felt like I might be dying, I was only 80% of the way there. So I still had 20% more to give. And I always was taken aback by this comment, and of course it's stuck with me for a long time. And being part of a community that wakes up at 5 in the morning meant that I slept through a lot of 8 a.m. classes. But I was able to manage this. This rowing community got me over that one hurdle, and now I was in a good place, and I got past the disappointment, and I was able to continue to work, and I worked everywhere, including at Lululemon during my undergrad, and I still have one of my good friends who I met through the rowing community as a big part of, an integral part of Lululemon. But then my father died. And I couldn't deal with it anymore. I couldn't deal with the financial instability. I couldn't concentrate. I didn't know what to do. And so I went to McGill and I said, can you help me out? Can you help me out financially? And McGill literally said these words to me, why would McGill want to waste their money on you? And I thought, I don't know, actually. So I quit. I quit school. I quit my undergrad, and I went somewhere I could get money so that I could actually survive. And it was really integral that I had met these people in different communities, because I ended up living with one of the people I played soccer with, and she was phenomenal. And without her, I wouldn't have kind of gotten through that, because she actually ended up spotting me a couple times on my rent and on other things, and was never pushing me to pay her back immediately. And maybe I haven't even paid her back all the money that I owe her, but we're still very good friends to this day. And I was, I always thought, okay, this is just temporary. I'll go back to school. I'll finish my undergrad. But I was terrified. I was really worried that I would be comfortable. I would be comfortable being, somebody who had money enough to pay their rent and buy their food. And that was better than trying to go back to school. Luckily, I wasn't very satisfied. I was quite bored. And about 6 months later, I did return to McGill and I finished my degree. But it was hard because I had lost my best friend, and that wasn't easy to get over. And so I took another break. And again, I was actually quite lucky, because I was able to find a job in the genomics industry, the genomics industry. And that was at a very pivotal moment in genomics because we were moving from low throughput sequencing to high throughput sequencing. And I got to experience that. I got to see the shift from regular Sanger sequencing to sequencing by synthesis to all of these other technologies that allowed us those breakthroughs that Joe talked about earlier, where we can get through entire genomes now in an afternoon. And it made me, I was able to learn a lot about technology and how technology can really advance the way that we can do our science. In that time, I decided to marry one of the rowers that I actually met during my experience. And I was starting to do well. I was feeling well. And I thought to myself, I'm going to go back to do that PhD I had planned on doing. And I'm going to, put my name out there and see what where I fit and what I can do. And I interviewed with somebody and it seemed like a good fit. And I started in the lab and I realized it wasn't a good fit. And it was very odd. And the requests that were being made and the context that I was being given, it didn't make sense to me, but I wasn't sure. And luckily I had a partner who could tell me, you know what, this isn't right. This isn't what you should expect. This isn't how you should be treated. And I think that's an important lesson as well, because sometimes when we're in academia or in these very kind of unique environments, we don't always have perspective of what's right and what's wrong. So I must admit that this restart into academia wasn't very great, but I was lucky. I was very lucky because it actually pushed me to a better lab. And I ended up joining a highly resourced lab that was very performant. And it wasn't easy. PhDs are never easy, but I was able to, have numerous papers during my PhD with a very good track record. And I was planning still. So I decided, because I was a bit older, having taken some time off of school, that this would be the time to have my kids. That way I could have a relatively uninterrupted postdoc. And so what I did is I had 2 kids in the last year and a half of my PhD, which, is a challenge, but I was convinced that it was the right decision. I was working toward my thesis. And right when I was set up with my good publications and my thesis that was actually awarded best PhD thesis in the department, I was ready to move away and my husband fell ill. So with 2 young children and no family support, I had to make a decision, which was to stay, to not move away. And of course, we all know, and you've heard from the experiences of the other 2 panelists, that it's important to move around, to really be able to learn from different environments. And this was simply not possible for me anymore. So again, my plans were a little bit uprooted. And in fact, I almost didn't even write that best thesis. But luckily I had an amazing mentor who wasn't my direct mentor, but somebody who'd always been a cheerleader for me. And he said to me, you know what? You're too close to stop now. Stop later. And I'm super happy and grateful to him that he pushed me toward that, toward finishing it. And then I had to work with what I had. And I couldn't go anywhere. I needed to stay where I was to take care of my family. And I was okay with that. But I needed to find a pivot, as Joe was mentioning some of the pivots that she found. And what I did was I searched for something that could be incredibly novel from what I was doing before. And that's where the single cell technologies came to me, and also EV research. So these types of technologies were really coming to the forefront, and I wanted to see how I could apply it and see how I could shift the paradigm in my own context so that I could actually make something of the trajectory that doesn't necessarily dictate the way that people are supposed to go in science. And like I said, this was not the path I initially envisioned, but I kept going because I had support. I had the support of my partner eventually when he felt was recovered, and the support from some mentors and the support from people that I had worked with along the way. And eventually I did get a bunch of high impact publications that allowed me to get an appointment. And it was interesting because the appointment finally came in April 2020, and we just heard from one of the other speakers that that was the height of the pandemic and literally everything was shut down. So while I had the appointment, I was actually not able to start my work or recruit students or do anything. But I mean, I don't know, at this point where my path will go, but I do know that if I stop, then my journey ends. And one of my close friends in academia who was a little more successful in following this traditional trajectory, once she shared some advice from me from her female mentor, which was sometimes you just need to ride the wave of opportunity. And I reflect on this a lot, on my past and future trajectory. And I also reflect on words that came from one of my rowing coaches who said, luck is directly proportional to the amount of work that you put into something. So sometimes I try to take a step back and I try to give myself a little bit of credit for the incredible luck that I've had over the years. And now I feel very, very lucky indeed to be working with brilliant trainees who share my vision and are eager to work toward answering the questions that we believe matter. And so you won't be surprised to know that there's a part of my lab that works on the beneficial effects of exercise on mental health. And really where I've ended up is a direct reflection of my journey in sports, neuroscience, psychology, molecular biology, genomics, and being a woman. But my interest in studying female biology doesn't come just from being a female. It's also because I have come to appreciate how little and how massively understudied females are in biomedical research. And the most salient example that I can give you from that is drug trials that were initially only done in males. And as a consequence, there have been a lot of dosaging, and it continues to occur, where we're dosing medication the same for males and females, and metabolism of these drugs is not the same between males and females, and it leads to adverse drug side effects and sometimes even death. And so Ambient is a salient example of this, that this was misdosed for 20 years, and it was only after numerous reports that they finally half the dosage. So it's clear to me that we need to disaggregate the way that we study female and males in science, and particularly in the brain. And so now what I'm trying to do is really understand how the individual cells of our brains are programmed by our sex-specific milieu, because of course we have many different hormonal concentrations and shifts and spikes throughout our development that influence how our cells are expressing different genes and how their programs progress through time. And I believe that that lays the framework for some of the pathologies that we later developed. So what I'm doing is I'm leveraging postmortem tissue, specialized animal models, single cell technologies, both in situ and in suspension, to do semi or pseudo-longitudinal profiling, where I can actually follow cells as they shift as a consequence of the hormonal milieu. So basically, the road is very linear for most people, I think, and perhaps mine was even less linear than most, but I think that we can still build on this if we just keep going. That's one of the things. And of course, to surround ourselves by people we know can be our champions. We still have a lot of work to do for women in STEM to make sure that we do get to those leadership positions, and that even when we get to those leadership positions, making sure that we get an equal salary. And I do want to point to this amazing group of individuals who now, I think, believe in the work that we're doing together. We're a diverse group, and we're working toward answering some of these questions about female-specific physiology and biology. So I'll end at that.
Thank you very much, Karina. That was amazing. It was a really incredible story to hear. And actually, I wanted to say that as you were going through your non-linear, very adaptive, very ever-changing journey, it reminded me of this Robert Burns poem that goes, "The best laid schemes of mice and men go aft astray." And it like totally exemplified that, but it was just so cool to see how you took every step and you changed with what you needed. And so yeah, it was amazing. So I think also, I wanted to say that the illustrations were very beautiful.
Oh, thank you for pointing that out. I need to credit Ren here for this. She's an amazing science communicator, and she helped me enormously with these slides. So thank you for pointing that out. She's fantastic. And
So, Yeah. So maybe we have time for one very quick question if anybody's got anything burning on their mind. Otherwise, we'll have lots of time. Oh, I already see Stephanie, if you want to unmute yourself and ask away.
So again, thank you for sharing your very inspiring journey. What I was wondering about, how did you find your mentor?
So it's interesting because I think he found me. So I was part of a relatively big group, and my actual mentor, who's Gustavo Turecki, was quite busy. And so it becomes difficult sometimes when you're in the highly resourced labs. Of course, you have, on the one hand, these this ability to be able to do all these things, and that can be really advantageous in terms of producing things. But then you tend to get a lot less direct mentorship. And this was another mentor who's kind of part of the group, and he was just absolutely fantastic in helping me. But I do have to say that now, I continue to follow this strategy, but which is to follow to group with people who are willing to be champions, right? Like who are willing to support you and tell you and remind you that the stuff that you're doing a good job, because often I'm not sure what success is. I have a hard time defining that. And so I need people telling me this. And so right now I have a group of early career researchers, and we're kind of going through it together, and we're telling each other, okay, you know what? That thing you did, actually, that you need credit for that. And we have to tell each other, because sometimes we don't know. And that kind of goes back to my first experience of trying to jump back into a PhD and not knowing that kind of environment that I was about to get into was wrong. And so it's really nice to try to find people you trust, that you can be open with and vulnerable. As I'm just vulnerable with about 29 people here. But it matters. It matters to try to find those people, and it doesn't have to be, it can be peer level. It doesn't have to be. I often don't even necessarily want that person who's going to tell me you should do this and this and this and this. Sometimes I just need the person who says, you know what, you're doing a great job. Sometimes that's just what you need. So you need a bit of all the mentorships. And if you're part of certain institutes, sometimes they set up those programs for you where you can be paired with a more senior individual, and that's super helpful. And you can maybe talk and see, is that person going to be more like, you need to do this, this, and this, or is that person going to be your cheerleader? And maybe you can have two. Maybe you can have a cheerleader and somebody who's going to kind of tell you the path you need to follow. Although, as you can see, I'm terrible at following paths. So.
Thank you.
Okay, thank you again. So we'll move on to our next speaker. It's a pleasure to introduce Dr. Adriana Suarez. So Adriana is a senior scientist and technology advisor at 10x Genomics. She holds a PhD in functional genomics from UBC, and she has over a decade of experience in biotech. So Adriana connects scientists with different innovative technologies in the spatial field, and she really helps them make the most out of these technologies to drive breakthroughs. And I can personally vouch for this because she's helped our lab a great deal over the years. And so she also contributes to different leadership networks which try to promote diversity and inclusion in STEM. And she comes with many different awards for her contributions to this. So without further ado, Adriana, I'll let you take it away.
Thank you, Ashley, for that kind introduction. It's really a pleasure being here today with sharing the stage basically with women that I admire and that inspire me. I don't have slides, and actually I'm glad I didn't prepare slides because Karina's and all the other presenters had amazing slides. So I thought, I feel that this year is the year of change, and there is so much change happening everywhere. So I do use a lot of slides in my work, so I thought let's just do something different for this presentation. And so I had like two main things that I wanted to share, and I think it's timely because of the questions that we got and the conversations we've had so far. I want to talk about mentors, and I want to talk about new technologies and what it's making me excited right now. As Ashley mentioned, I'm a science and technology advisor, so it's an interesting role because I'm in the intersection of technology and all these new products that 10x Genomics develops. So I'm always learning on the technology side. But then I'm also really close to the science and the applications, basically how these tools are being used in so many fields, from neuroscience, as we saw today, to oncology and developmental biology and many more. So I'm in this interesting space where I need to keep up with the technology and also with the applications that are being enabled by these new products. So within this role, I do a lot of education, also relationship building with key opinion leaders to really take these tools and help these tools really reach the impact that they can have in all these different fields. So it gets busy. It's a lot of things and a lot of moving parts at the same time. And this is where I think your mentors and your network helps you thrive, or at least for me. So I wanted to highlight 3 mentors that I've had in my life. I mean, I thought about this and I wanted to pick 3, and it was pretty easy. Actually, they came up quickly. The first mentor actually came very, very early. I don't even know if she knows me. And it really came when I was finishing my high school. So I'm from Colombia, and there you have to do this really hard exam when you finish high school to get into university. So you really want a high score for that. So I joined this intense workshop kind of course to be able to get that high score. And it happened that the science part was being taught by a scientist. So it was the first time that I was exposed to what a scientist will do. And she came and explained, this is how I will design an experiment. And it happened that she was working in genetics. So it was love at first sight in terms of genetics and omics in general. And so since then, I have been really passionate about how all these tools could be used in so many different ways. And it really drove my appetite to do research in different areas and move around that space. Another very important mentor for me that I'm lucky enough that is a really close friend of mine really came later, like maybe 5 years ago. She's from Colombia as well, and she works in funding agencies, leading a lot of projects to basically bring access to genomics technologies. So in the South, so in developing countries as well as here, she's a survivor for cancer. She is such an energizer for me, just seeing all the work that she does to move barriers, jump challenges. The energy she has is just contagious. So I think having her around me has been super special. There was a question about how you meet your mentors, and it happens through networking. I really I stalk people. Basically, I go on LinkedIn, and if I see something that I share or that it could be potential synergies with a person, I will connect with them. And I think some people do that as well. So I do that a lot. I used to go more to networking events when I was in grad school and when I was starting my career in biotech. I do that less, but I use different events, conferences to basically meet new people and find those synergies. I have a friend that she says, I mean, in networking, you have to kiss a lot of frogs to finally find your princess or your prince. And I think that's true. Not because, I mean, there is something wrong with other people. It's just that you're going to connect with people in different ways. And a lot of these relationships are going to happen organically. So to Corina's point, to have some of that luck to find your people, you want to put yourself out there, network, and build those relationships. The last person I wanted to highlight is actually my boss, my manager here at 10x Genomics. He has been such an inspiration for me. He has a really interesting leadership style of combining empathy. So he cares for you, but then he also cares about the impact that these tools have, the impact at the business level in our company. So he really matches these priorities with your strengths and then helps you thrive, which is quite amazing, right? So I'm very grateful for having such a leader that allows me to explore ideas, lead ideas, start initiatives, fail and go back to it and try other things with guidance, right? So it's important to keep that focus and to have that direction. So you keep that path somehow aligned with what's important in the long term. So yeah, I mean, those are three important people in my life. It's a combination of mentors, champions, allies, And so it's really important that we have those, and they are also representing different communities and different fields. So you have that help, because when you are developing your career, you're going to need help. And if you don't ask for help, you don't get it. So really putting yourself out there, asking, telling people what you want. And this was another advice actually from one mentor that I had. It's just, you need to ask for help. I was in a job at some point and I wanted to change jobs, but I just felt fear of accepting that I wanted to change. So I had a mentor telling me, just ask for it, share. So I think that's an important part. Okay, so the other part that I wanted to talk about is it's on the technology side, more on my work side of things. So at the Next Genomics, we design and put together different tools to do different things. So single cell, spatial. And I think if you are working in this space, as you heard today, you are in the right place at the right time. We are in this special time where we have these developments at the technology side. So we have like tools like Xenium, right, that allows you to get subcellular resolution of transcripts from tissues directly with thousands of genes at once. And then you can add protein as well. So it's just like mind blowing, the amount of information and the impact this could have in so many different layers. But then you also have this amount of data that you need to be able to analyze and find trends. So we are seeing the development of these LLMs and all these machine learning models that are allowing us to understand the trends behind all these data that is being produced. And so there are a bunch of initiatives in terms of single cell with atlases that are being developed. Chancellor Conveyor Initiative has one, the ARC Institute has another one. And then there are also initiatives on the spatial side where basically people are generating data sets on Xenium, for example, and then using that data to train LLMs and then make predictions, right? So in the future, you can imagine how with a simple stain, you can predict what's going to happen. So I think it's, again, an exciting time because we have the tools on the technology side, but also the data analysis side. And so I think thinking about those topics, it's very important. And as we continue to move forward with these technologies, these two, the combination of these two, AI and single cell and spatial, are becoming very important. I'm fascinated by AI in so many different ways. Like you have these LLMs that are being trained with data, but also you have AI for your own life and your own development. So in data analysis, we're actually running an initiative at 10x Genomics where we guide people on how to use AI for data analysis. So we have some new conversational tools that allow you to basically, it's a chatbot kind of a style where you just say, hey, I need to analyze this data, guide me through it. So there are cool agentic kind of workflows that we can use to do that. So it basically lowers the barrier for non-computational biologists, like people that do not code, but then have that biological experience and knowledge that is so fundamental to analyze this data. So all of these topics are top of mind for me. So if you want to chat about this, or if you have questions, happy to connect. Happy to take any questions, or we can wait until the end.
Yeah, wonderful. Thank you so much. Does anyone have any questions? We can maybe take one before we move on to the next portion. All right. Thank you again. We'll take more questions during the panel discussion then. And it is my pleasure to introduce our last speaker for today, Dr. Adunola Adebi. She holds a degree in veterinary medicine and is transitioned into spatial biology about 3 years ago. She's currently a PhD candidate at Stellenbosch University in South Africa, where she investigates the immune landscape of TB lesions using single-cell and spatial transcriptomics. Her research explores how immune interactions within TB granulomas shape disease progression and treatment outcomes. Driven by her passion for knowledge sharing and grassroots empowerment, Dr. Adebi co-founded BioInform Her, which is a platform that helps women from underrepresented communities gain bioinformatics skills by simplifying complex concepts and providing mentorship. Over the past 2 years, BioInform Her has supported hundreds of women across the globe to begin their journey in bioinformatics and computational biology. She believes that innovation in science should not be limited by geography, and through both her research and advocacy, she continues to build capacity for spatial and single-cell biology in Africa and create space for more diverse voices in global health research. Please take it away.
Thanks so much for introducing me. And hi, everyone. My name is Adebi Sefiat Odola. And just as she has said, I'm a PhD student at Stellenbosch University here in South Africa. I think everybody have like very interesting story. I'm literally new in this field, so my story is very short. Well, I'm very sure that you will still be inspired. So yeah. Let me just a minute. Okay. So I have my first degree at University of Ibadan in Nigeria. So I'm originally from Nigeria. And I actually discovered bioinformatics in my 5th year. So the program is actually a 6-year program. And in my 5th year, I came across these African Bioinformatics Network Fellowship. And I was like, I was bored already in vet school. I wanted to try new things. And I was like, okay, there is no harm in doing some programming. That was what I call it then. So I decided to opt in for these African Bioinformatics Network Fellowship that year. And to my surprise, I got in. I didn't even know that I will be selected. So I was selected, and it was quite intense, difficult. I didn't understand a lot of things there, but I love the fact that I was learning something totally different from vets, totally different from taking care of animal. Okay, I come back from clinics and I could do something totally different. I love math a lot. So it's kind of very interesting to me. So I kept doing it and I was able to complete the fellowship. Afterward, I also came across H3 Bionet. It's actually a 3-month fellowship too. It was run for about, I think, roughly 10 years. I think the program ended last year. Yeah, so I did that, but those two programs were both on genomics. So my first experience for transcriptomy was a summer school I did at RNA Institute at University at Albany. That was in 2022. Yeah, that was it. I actually get to really understand bioinformatics because the summer school was really intense and was really we had a lot of mentors that you could easily talk to during the summer school, and they kind of guided us through the whole process. So it was really interesting for me. And since then, I started like reading on my own. So I decided to pick R as my programming language because I found Python quite kind of difficult for me during that time. So I picked R as my programming language. So from then I started studying on my own, read textbook, go through a lot of what's the name, a lot of YouTube videos. But I mean, one of the best textbook that I really love when it comes to R is R for Dummies. When anybody asks me about learning R, I always recommend that because it's so easy for you to understand. So I really enjoy reading that book. So after I finished vet school, the year I finished vet school, we have a program in my country. It's a community service. You have to give back to the community. So you have to spend 1 year to actually serve in any part of the country to kind of pay back to your country for kind of sponsoring you. Yeah. So during my community year service, I was posted into the northern part of my country. So I was posted to work at National Veterinary Research Institute. And that was where I had my first experience of research, in-depth research. And in that lab is actually a reference lab. So we always have a lot of sample from different parts of the country, different animal sample, mentioning pigs, what's the name, bells, different sample, wide animal sample. So I get to like work with different type of sample, fecal tissues. And what I was majorly doing that time is I was molecular biology, genomics. So I was doing RNA, DNA extraction, then PCR, different type of PCR, both what's the name, qPCR and the normal PCR. And I had a very little bioinformatics there. But it was just so interesting for me to fully involve myself in research, and I kind of grabbed the whole concept of how research goes. It's not like what we see when we read paper. It's not as easy as we just think people go into the lab and they just come out with the result. I got to see the whole process that goes into the work that people actually publish. So it was quite interesting to me. But as much as I enjoyed the wet lab, I kind of find the data analysis part more interesting to me than the wet lab work. So I already had it at the back of my mind that yes, it's possible that I will likely be doing dry lab work, because for me, it's very flexible and I just like something that make me think very deep. And to me, dry lab allowed me to do that. So in 2023, I applied for an MSc opportunity, sorry, at Stellenbosch University in South Africa. So I'm currently in South Africa doing my PhD. So I applied for that MSc. The MSc is in molecular biology. But when I got here, I was told I was going to work on TB. I was told I was going to do spatial biology. But when we say spatial biology, I was just thinking of, okay, I was going to do some histology work with immunofluorescence. I mean, that is not really a big deal. But when I got in, I was told that, okay, you're going to be doing spatial transcriptomy. And I was like, what? I know transcriptomy, but spatial is too advanced for me. And I was so scared. Funny enough, in the whole country, there were just two labs that were doing this spatial transcriptomy. Our lab and another lab in Durban. Durban is like a lot like 2 cities away from Cape Town. So I'm in Cape Town. So Durban is far away. And this technology is relatively new in the two lab. Funny enough, the technology was also new in my lab. Like I was literally the one that pioneered this technology. So I didn't have anybody to talk to when it comes to my analysis, although I had help with running the bench work, and that one went on well. But then immediately I got my data, my sequence data, I had problem with it because there was nobody to talk to. I was kind of stuck. But the interesting thing is I have amazing supervisors, like amazing. And I mean it, I'm not overhyping them. They are very amazing. So I was initially scared, but then because of the support I got from my supervisor, it began to make sense. So what they did is they started connecting me with experts abroad, like uh, special bioinformatician that specialize in spatial transcriptomy. So I got to talk to them. I got to ask them questions. Anytime I'm stuck, I will email them, then wait for like 2 or 3 days for them to reply. They recommend paper to me. They walk through my data and started making sense to me. So just to show you what I'm working on looks like. So literally I'm looking at tuberculosis. So we have a lot of cases of tuberculosis in Africa. We also have it in Asia, but South Africa and Nigeria are like the leading country when it comes to TB. So I'm actually looking at TB in children. One of the common manifestation of TB in children is extrapulmonary TB, and it usually affects these lymph node just under the chin. So sometimes you find that the lymph node here becomes swollen. So when they become swollen in children, they could swell to the extent that they will press on the airway. And once they start pressing on the airway, there is a need to take them out. So they always undergo surgery. So once they undergo surgery, I mean, they get the tissue out and they stitch the patient up. Once they stitch the patient up, the tissue become like it doesn't have any usefulness, but then they don't throw it away. So they actually, what they do is just embed it into a paraffin wax block. So we have like a large archive of these TB tissue. This lesion is actually called granuloma. So this is what it looks like histologically. And from literature, we know what granuloma is composed: macrophage, some immune cell macrophage, T cells, Then we have fibroblasts. That's what you're seeing here. I think I need to use my cursor. Just a minute. Okay, that's not going. Sorry, this is not okay. So you have some fibroblasts here, then you have your macrophage here. So that is what it looks like histologically. I'm very familiar with the histological aspect. I'm a vet, I'm very good with that aspect. It wasn't a problem, but it became a problem once I was told that I have to like start looking for molecule combine combine transcriptomy with the histology and start looking for what's the name? Start looking for what molecule these cells are producing, how they are communicating with each other, how they are relating with each other. So graphically, this is what we expect that a TB granuloma will look like. You have your TB, the pathogen. Those are the one in red. You have them, we call them mycobacteria. You have them at the center. Then you have this immune cell, which are macrophages, but they have been elongated. So they are epithelial cell, but they are macrophages that actually develop into epithelioid-like cell. Then you have the T cells outside. Then you have the fibroblasts. That is what we see in this histology. Now, what I work on for spatial transcriptomy is looking at the same histology, but now overlay with sequence data. So I look at these different cells, where they are, how they are communicating. It was very difficult at first, but now I'm very good with it because of different support that I got. So working with those experts abroad, one thing that they did for me is that they allowed me to join their lab meeting every week. So I get to listen to different people from different lab present. So I get to understand things from different perspectives. If I've tried this method, maybe to do pathway analysis, maybe to rank gene for my GCR analysis, gene enrichment analysis, then somebody can come and say, oh, I use this other ranking method. Maybe I use moderated statistics to do my ranking and I got something better. Then I go ahead, read more about the moderated statistic and understand how it works. Then I try it on my data and it works. So joining the meeting really helped me. I started learning how to do my analysis better, even how to present better. And this mentorship from the expert actually changed how I learn. I no longer just scroll through papers. I take my time to read in depth, go to the supplementary part, try to understand, try to criticize what the paper is saying. I don't take whatever the paper says for what it is. I try to reason deep. So slowly I began to see pattern. I began to gain confidence in my analysis and make progress. And one thing about me is that I became so curious about this spatial transcriptomy that I always want to talk to people about my project. Once I say that you are somebody that is interested in research or interested in something related to bioinformatics or something related to TB, maybe you're a PI or you have your own research that is similar, I'll ask you, oh, do you know about this? Do you want me to show you my data in as much as my PI are okay with it? Then I'm always eager to share my work with people. And by sharing my work with people, I gain a lot of feedback and that helped me to improve my work, to improve and even directed me to what I wouldn't have thought about if I've been so quiet. I've been the kind of person that doesn't talk to people. And another thing that helped me, I had an opportunity to do a research visit, a 3-month research visit to Germany last year in 2024, sponsored by DAD, my founders. So I worked at Professor Anka's lab. This is Professor Anka A, and I got to work closely with a bioinformatician, that's Dr. Sebastian. And working with him makes things easier for me because anytime I get stuck, I could easily say, Sebastian, I'm stuck here. What do you think is the problem? Then we troubleshoot together. And that really helped me. So that helped me to realize that science, but not only science, even bioinformatics or any analysis that you're doing, it grows faster. Things become easier when you share and collaborate. So once we are talking about data analysis or we are talking about science, we shouldn't see ourselves as I can do everything. We should always see ways to collaborate with people, ways to share things with people, and they don't have to be in your field. They don't have to be doing TB. They don't have to be doing multiple sclerosis, but they might be using the same tools that you're using. So that's an opportunity to collaborate and see where you can actually work together. So having seen the struggle I passed through to actually become so good in spatial transcriptomy and bioinformatics as a whole, I realized something, that I'm not the only one passing through this struggle. We don't have a lot of bioinformatician in my lab, and we only have few students that are actually even doing bioinformatics analysis, like bioinformatics analysis based projects. So I realized that I'm not the only one passing through this struggle. And if there is any way I could help people, I shouldn't be the only one to benefit from this support I got from my PI because they connected me to people. They can't connect everyone to people outside the country. So can I, in a way, give back to my community? So I am my friend is in Canada. He's also doing a little bit of bioinformatics, and he was planning to do spatial transcriptomy, but then he couldn't continue at some point. Is at Manitoba right now. So we came up with the idea of bioinforma in 2024 February. And the whole idea is to actually teach people bioinformatics, but in a very simple way. One thing about learning bioinformatics is that when you ask people randomly, especially people from developing country like ours, where we don't have the opportunity to learn all these programming skills while we are in school, when you ask them, they'll be like, oh, coding is hard, I don't want to try it. And when you ask women, they'll be like, those are for guys. Those are very hard things. You shouldn't do it. You should think of becoming a doctor or becoming a nurse. So they think it's hard because they want to do the big thing immediately. They don't understand the small thing. So we think the easiest way to help people to realize that this is not really difficult as you think is to make it simple, make it relatable, and make it accessible for them. So we came up with the bioinforma idea in 2024 February. And since then, we've been running our program every week and literally every month. And about 200 women have benefited from our different program across the globe. But mostly we always have people from Asia and Africa that participate most, but sometimes we have some people from Canada, like Black women from Canada that participate in our program. So far, few of the programs that we've hosted involve my story in bioinformatics, where we bring females that have actually overcome the same barrier of having to navigate into bioinformatics, especially from other fields, to come and actually tell their story. We had Shamini like last week, I think so. Yeah, she came to share her story with people, and people get to see that, yes, you can do it, even if you have a degree in something totally different from bioinformatics, even if it's in English or literature and you're interested in bioinformatics, it's possible in as much as you have a goal, you see where you're going to use it. Yeah. We also have another program where what we call Get Up with GitHub. The whole idea is for people to document their bioinformatics journey. So people will tell you, oh, I'm into bioinformatics, then show me the proof. They can't present anything. So we think it's very important that people know how they should document the journey. So we came up with this program, and we had a lot of turn up, like people were willing to learn, and a lot of our students have like good GitHub profile. So some of our projects that we always run every week, not project, we call it tasks. We always have like short tasks. And at the end of like a module, we always have a mini project where we combine everything that we've taught them. So one of it is this module 2 mini project, where we actually had a short project for people to actually participate in. And out of people that participated, we selected about 40 people who we gave scholarship. We actually sponsored them, and we did a 8-weekend art programming. It's actually basic art programming for them. And I completed that in September, end of September. And we had some of our students they presented their work last weekend. Yeah, so you can check our page on LinkedIn Yeah, we have about 8,000 people that follow our programs on LinkedIn. So you can check us out. And if you're interested in our program, you can always like join us anytime we are having our interesting program. So one thing I would like to say is that I've been in Africa my whole life. Yeah. And one thing I would say is that we have endless possibility in Africa, especially when it comes to spatial biology. This field is relatively new in this continent, even though more lab, more research group are now getting interested in it. The main problem that we have now is not only the fact that it's expensive, yeah, it's expensive, but people are finding a way to get into spatial biology. But the thing is, we have a wide skill gap, like very, very wide. I've been there for the past three years, and still we don't really have a lot of people that are very good when it comes to spatial transcriptomy. And now we are having like different type of spatial transcriptomy platform coming up. So we need more people to actually comes into this field. And one thing is, it will be easier if there is a global community where people can easily learn, share, and grow together. I mean, learning on your own sometimes is not as easy as when you can learn alongside other people. So for me, I always want to encourage people. It's not that difficult. You can try it. You can go for it. If you fail, you try it again until you get it. Yeah. So that is something I always hold on. If I do not do well today, if my code did not work perfectly today, I'm very sure it will work well tomorrow. Yeah, and that has been helping me. And another thing that has helped me in this my what's the name? This is my bioinformatics journey, and currently in my PhD program is that I'm always curious. I want to try new things. I'll tell my supervisor, what do you think, Nelita? Do you mind if I run this? And she's always willing to let me try new things. And she's always ready to like work with me when I'm showing her my result, when I'm explaining my result to her. So having somebody that is very supportive to it is very important. But you being passionate, being curious about what you're doing is something that you can discard. If you are not passionate, nobody will be willing to help you. And of course, seek mentorship. Mentorship does not have to be like you have to be on the person's neck or you have to be taking their time all the time. You can just be shadowing them. It's a form of mentorship. You can just talk to them once in a while when you have a serious problem. You don't have to be having regular meetings with them. Yeah. So yeah, so basically that's my story. It's very brief, but I have learned a lot along along this journey, and I've had the opportunity to like meet a lot of people and present my findings in different different conference, both internationally and locally. Yeah, recently I presented my work at the TB Immunology Union in September. Yeah, and very soon I'm going to be presenting it again at TB Union in November. So it gives me joy when I meet people and people say, oh, we are into something like that. Then like, can we talk about it? Then we get to talk about it and we get to work together. So we've been collaborating with people on different aspects of this work because yeah, I'm willing to do that. I'm willing to work and I have somebody that is willing to support me. So I would like to say a vote of thanks to the following people. Prof. Nandita Duplessis, Prof. Garawazi, those are my amazing supervisors. Like they are extremely amazing. And Dr. Olanipe Kunduatobiloba, that's the co-founder of Bioinforma. Dr. Lo Shola Olaoluwa, that's my supervisor when I was at National Veterinary Research Institute when I was doing a lot of PCR work. Then I have JW. JW is the expert that I work with, is actually in Harvard University. Then I have Prof. Anka from Germany. She hosted me, and anytime she meets me at the conference, she's always like, come back for your analysis. I still have bioinformatician. You guys can still work together. She always want me to come back. Then I have Dr. Sebastian. Dr. Sebastian worked with Professor Anka, and she is actually the bioinformatician in a lab, and we worked together when I was there. And the RRJ team, which is actually my the research team which I belong here in South Africa. I would like to say thank you to all of them, and thank you for listening to my little talk. Yeah.
Fantastic story. Thank you so much for sharing. We're really happy to hear about it. Very inspiring. To be mindful of time, we have posted a link to Slido, which is our platform for questions. We would like to ask the audience to take a brief moment to vote on the questions and to be mindful of everyone's time. We'll take the top 3 for the panel discussion. Okay, I see the question on self-doubt is gaining lots of popularity. So many of us have experienced self-doubt. How have you learned to recognize and manage imposter syndrome, especially when surrounded by male peers or leaders? Would any one of the panelists like to share something on this topic?
I can start. I will say that it is something that you go through it. I mean, there is the imposter syndrome. It's called different ways, but I think it's so important that we reflect on our strengths. And something that has been valuable for me is to stop comparing myself with other people, because your journey is so unique, so it doesn't really make sense. And then play with your strengths. Uh, I mean, I meet a lot of trainees and students in my daily day, and how many times I've heard, "Oh, I'm just an undergrad. Oh, I'm just a master's. Oh, I'm just a PhD." So never start your introductions with "I'm just." You are who you are, and you own it. And whatever is your experience and your level, you need to craft an elevator speech that really represents you when you have these interactions. And it really helps you build that self-confidence, because as you build that elevator speech, or your kind of introductory paragraph to yourself, you reflect on what you've done, the initiatives you've been part of. Maybe now you're thinking, "Oh, I don't have this, or I don't have that, I don't have a paper, or I haven't been part of that." Well, you can start. And so your elevator speech could be, "Hey, this is me and I want to get here." And so it could be more inspirational and goal oriented than like a past experience. So I think that's something that has helped me, and could help you. So working on a 1-2 minute kind of introductory presentation paragraph could help.
Yeah, and I'll just add this, it's a female challenge. Men don't seem to have as much self-doubt. And so I think it's kind of innate in our biology. I don't know, Karina could comment on that a little bit more, but I think we need to realize that men have been faking it till they make it, all this time, and they get in positions of leadership. So I think we just need to be confident, like Adriana was saying, that we're just as good and we are just as smart, and we should have less self-doubt because we're similar. Karina, do you know from a biology standpoint?
Not yet. I have not found the gene that encodes imposter syndrome. But I just want to piggyback on everything that you guys are saying, which is that, all of our trajectories, our journeys are different. And, I know that from, very significant experience. But sometimes, you just need to take a step back and give yourself context. And I agree that, don't compare yourself to other people. just define what it is you think is success, and then take a second. Did you reach that thing that you thought you were going to reach? And in most of our cases, we're there, but we don't want to give ourselves the credit. And that's the thing that's happening. So I think if you take a step back and you just really try to contextualize. That being said, I think there's nothing wrong that we always want more. And that's something, I want more. I want to make sure that this lab continues to grow, that I can continue to support them. But, up until now, the things that I've done, it has been a success. And so, give yourself that credit. And those types of check-ins, I think they're really important. And yeah, don't compare yourself to John Doe over there. Just, compare yourself to what your standards are. I think it helps.
Wonderful. Thank you so much. The next question is, in what ways do you think diversity in teams leads to better tools or methods in spatial and single cell biology?
I truly believe in diversity. Like every mind is different, and every mind has an asset in the whole process of science. So some people might be more analytical, some are more mathematical. It's not sex-based, but just different minds are good at different things, and that's the reality. And so we have to embrace, I think as a person running my own lab now, it's our job to get the best out of each one of our people in our lab. And when there's a challenging person, I don't think of it as a challenge. I think of it more as, we haven't found the right spot for you. Let's work on this. Let's find the right angle that makes you shine. And so I really try to instill confidence in my people and allow them to realize they do have a role, and we need it. Like, dry and wet lab people are absolutely essential for the types of labs that we all run. Mm. That's it.
Thank you. Let's take one more question. Women often have or wish to have children and have family as a priority. Do you have experience of balancing this with a scientific career? So I know some of the speakers already shared their family stories, but is there something you would like to add? Any advice?
I can go on that. So I had my son when I was in the second year of my postdoc, and I took a 6 months break. I think you learn to prioritize things when that's the one thing I've understood. And Frida's, my mentor, has told me that you have to pick the priorities, and there are some experiments that can wait, and the others have to be done. And then you might not have the same amount of time to socialize. You can work and then go back home to have some family time. That's okay. That's just a phase of life. Um, but yes, prioritizing my things is something that I understood, and I'm using my time more wisely now. I was basically a lazy person, but now I don't have the time to be lazy.
Yeah, I agree. I think that there's a lot of prioritizing, and there's a lot of just good use of your time. Um, I can't say that I have any free time, but it's okay because, I guess what was free time, I use it differently. Like, I very much enjoy being with my family. One of the things that I do, I suppose, in my free time is I coach my kids in soccer. So this is a theme that continues, and it's something I get to give back to them, to both of my kids, and it's something that, keeps me going. So I think that, kind of bringing them into your reality. I also do a thing, and I'm not sure that I would recommend this for everybody because not everybody has the same type of kids, but I bring my daughter often with me to conferences, and it's an amazing one-on-one time that I can have with her, but she's also very independent, so not everybody can do that. But I really enjoy being able to do that and being able to integrate her. And so I I know that there's a lot of people who want to have defined sort of sort of compartments in every aspect of their life, but I actually kind of just mix it all together. And I bring my kids in, and they, everybody at the lab knows my kids, and bring them places, and I, hope that they like the things I like so that we can enjoy them together. And so there's this whole thing, and it's just one entity, but it allows me to manage a little bit better the fact that I really want to spend time with my family, but I also have a lot of commitments and a lot of time that I need to spend in this particular position. So this is kind of how I've come to balance it. I don't know that it works for everyone, but I will also say that I would never say that you should concede or give up this family life because you think you can't have that and be in this position. I think you'll find a way that you can manage it. It's unfortunate that we even have to think that we might have to concede that, but you will figure it out.
Yeah, I'd like to just emphasize, do it. If you want a family,
Exactly.
please, 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 it'll work out. It
Yeah.
will. And I think that'll all go away, all the hesitation once it'll work out. Life
Mhm.
has a funny way of working out. So do it all. We shouldn't be limited just because of our biology and we're the ones that have to carry a baby.
Mhm.
There's ways to make it work and balance well with your partner, etc. Yeah.
All right. I think, Ken, all right. I think that kind of comes to the end of our event. Honestly, it just flowed. We didn't even control anything, and we have all of you to thank. So before we end, I wanted to share something. So when we started, I didn't want to say how we still need equity, but we do. I don't want to open up the session saying that. I opened up saying that it's a celebration, but the reality is that we haven't really met or hit that equity. So maybe I just want to end by saying something that my grandmother shared with me. And my grandmother is, she played a, she still plays a huge role in my life. And I remember when I got my PhD, and there were so many around me who were telling me that, oh, you should be getting married. You shouldn't be taking the trip to Canada to do your PhD, so on and so forth. So I was teasing my grandmother. So I said, secretly, you want me to get married and just stay here with you, right? And then she became serious and she said that, yes, watching you as a bride would make me happy, but watching you live your dreams would actually allow my soul to actually rest in peace for all eternity. Because my grandmother was, she went through World War II. She wanted to study, she couldn't. She got married to someone who was twice her age, who was already married. So for her and for my mother, what we are doing, not just me, what each and every one of you is doing, you are actually breaking generational trauma, right? So that's what we are doing. We're actually giving something to posterity so that their lives will be better. So I think that is what this whole Women in Spatial is all about. And that is what the Inspire Me series is all about. And I really cannot thank each and every one of you, especially to Alyona and Ashley for like coming together and putting this together. And to the rest of you for taking the time and spending the time with us. So yeah, thank you so much for coming. So with that, I think everyone deserves a nice evening. So have a nice evening, and we will post this recording online as soon as we have it. Thank you so much.
Thank you.
Thank
Thanks,
Alright.
everyone.
you. Bye.
Thank
Bye.
you. Bye.